slide. We've gone over this a couple times for anyone who's new to this seminar series. We're really looking to hone in on the things that we need to consider for effector function. And this is effector function not only for monoclonal antibodies, which is usually a way we think about this, but also polyclonal vaccinal responses and making sure that we're inducing the right types of antibodies when we're vaccinating. And so the goal is really to, as Joe had outlined, review methods, data, conclusions, how effector function modulates different responses, and then to collate all that data into one sort of overall overview of effector function across the board. So if we pop to the next slide. All right, and so there's two groups that we've assembled, and you can see the rockstar lineup that are in both. One of the groups, which is headed up by Annie Zumsteg, who happens to be sitting in the room with me actually right now, is really looking at standardization of effector function assays with the goal of overall understanding how things really integrate and how we can move some of these assays to making clinical decisions maybe sometime in the near future. And then the second group is headed up by Galit Alter, who unfortunately couldn't make it today, but she is heading up the group that's been overseeing the seminar series. So on the next slide, I think we just have an overview. Mark's here today. Super excited to have him here to talk about microenvironments and optimizing antibody therapy. And then we have one additional seminar that Folk is giving in a couple of weeks on the 23rd, looking at effector cells in different compartments and disease states. So stay tuned, and hopefully everyone will be able to make that one as well. Right. And so Mark right now is a professor of experimental cancer biology in the antibody and vaccine group at the Center for Cancer Immunology, Cancer Sciences at the University of Southampton. He did his Ph.D. at Southampton and actually also completed some placements during his undergrad at Glaxo and Northeastern. And following his Ph.D., was actually awarded a career track fellowship with the School of Medicine and obtained a fellowship with the Leukemia Research Fund, which is now actually Blood Cancer UK, and completed some studies in the world-class laboratory of Andreas Strasser at the Walter and Eliza Hall Institute in Melbourne. He actually returned to the UK in 2007 to establish his own research group. And his current research really focuses on two main areas. So antibodies and small molecule inhibitors overall, but aiming to understand how these therapeutics actually function to kill tumor cells and how we can actually improve upon them. So I'd like to turn it over to Mark, who will hopefully be able to project his slides. And as Joe mentioned, please add any questions you have in the chat, and then we can have a Q&A period at the end.
Okay, so hopefully you can see those slides.
Yep, they look great, Mark. Thank you.
Fantastic. So thanks for the introduction, Lisa. So yeah, what I'm going to try and do over the next 45 minutes or so is really overview what we think we've learned about suppressive microenvironments and how they how they impact effector functions, and really talk through two papers that we've recently published. So, where did my slides not want to go? Okay, so what I'm going to cover really is some really basic background to start with about how antibodies delete target cells, talk about Fc gamma receptors, and then focus, I guess, a reasonable proportion of the talk on how the tumor microenvironment can impact the deletion of those target cells, and particularly by changing Fc gamma receptor expression, and particularly the inhibitory Fc gamma receptor 2b. And then the second part of the talk, I'm going to talk about how 2b actually impairs antibody-mediated deletion, and talk explicitly around the role of the ITIM motif there, and consider how best to block the inhibitory receptor with monoclonal antibodies. So I think the conclusion of a lot of people's work over the last 30 years or so really is that the antibodies work through Fc gamma receptors, IgG antibodies at least. And this slide really just summarizes the background in terms of the human Fc receptor expression profile and receptors that are available. So we have 5 potential activating Fc receptors and a single inhibitory receptor, Fc gamma receptor 2b or CD32b. And I'll no doubt interchangeably use those terms, so just watch out for that. The activating Fc receptors pretty much signal through an ITAM motif, and that'll be important later, whereas the inhibitory has an ITIM motif, so an inhibitory motif. And then the bottom part of the slide really just serves to exemplify the fact that these Fc gamma receptors are expressed on many cells in the immune system. And so the interaction of antibodies with the immune system can be quite complex depending on the expression profile of those different Fc receptors. And this only includes immune cells. There are also other cell types in the body that can also express different types of Fc gamma receptors. And we spend a lot of time trying to dissect mechanism, which typically means using transgenic knock-in or knockout mice. Unfortunately, the parallel system in mice is highly related. So we have a similar profile in as much as we have a number of activating Fc receptors, again, that signal through the ITAM motif, and a single inhibitory receptor, which contains an ITIM motif. One of the nice things about the mouse system is that they all require this common gamma chain for expression and signaling. So if you make a gamma chain knockout mouse, you knock out the activating Fc receptors and leave just the inhibitory. And work that we and others have contributed to over the years really shows that antibody isotype is really important for delineating deletion using antibodies in vivo. And we spent a long time looking at CD20 as a target and deleting B cells, normal, malignant, or otherwise. And this slide really just summarizes an experiment that recapitulates exactly the same as other people have shown for other antigens and other cell types. And that is essentially that in the mouse system, the mouse IgG1, shown here in the red circles, doesn't delete the B cells as effectively in a systemic system where you just give a single bolus of antibody on day 0, and doesn't ever delete them fully. Whereas if you use exactly the same antibody, so this is a version of rituximab deleting human CD20 transgenic B cells in the mouse. If you just convert the isotype from a mouse 1 to a mouse 2A, you get much more profound deletion, and that deletion persists for much longer. And we and others have contributed to the idea, and that it's really the interaction with Fc receptors that is mediating that isotype dependence. And it was formalized that the relative engagement of the activating to inhibitory receptors is really what's determining the level of deletion. So mouse IgG1 actually only interacts with a single activating Fc receptor and the inhibitory, so it has a low AI ratio, whereas the mouse IgG2A interacts with all of the activating Fc receptors in the mouse, and to a lesser extent with the inhibitory, so it has a high AI ratio. And that describes why it's good at deleting, whereas the mouse IgG1 isn't. And this paradigm exists not only for just deleting normal circulating peripheral B cells, but also malignant B cells. This is a lymphoma model, same kind of principle, taking exactly the same antibody with the same Fab regions and just changing the isotype to a mouse 2A or a mouse 1. And what you see very clearly is that the mouse 2A antibody gives long-lived cures to the majority of mice, whereas the mouse IgG1, all of the mice succumb to their tumor. We've also expanded this paradigm to other cell types, not just CD20 and not just B cells. And so we've shown similarly for regulatory T cells, trying to delete those. A mouse 1 doesn't delete them effectively, and may even agonize the expansion of those cells. And that's a different story for a different day. But the mouse 2A isotype is good at deleting those Tregs. And that can have impacts for different Treg markers, such as OX40. Here you can see in the green, which is right at the bottom of your screen, the mouse 2A OX40 is able to control the tumor, whereas the mouse 1 isotype doesn't do much above control. So the paradigm really is good in terms of deletion. And then the question becomes, okay, if the isotype is important, what are the other parameters that are important? Which cells, which effector cells, and which mechanisms are important for that deletion? And to address this, over 10 years ago now, we took a an adoptive transfer model system, which we've used a lot in the lab as a workhorse. And the system is detailed at the bottom here. So what we do is we just take target human CD20 transgenic B cells, label them with high levels of CFSE, mix them in a 1-to-1 ratio with non-target B cells that don't have the human CD20 transgene, and label those with low levels of CFSE, transfer those into a recipient mouse, give them a day to circulate into the spleen, give a therapeutic antibody or a isotype control, and then a day later, harvest the spleen and look for the deletion. And when we do that, it allows us to provide a target to non-target deletion ratio, which you can see here. And again, using the clinically related antibody that has been murine for use in the mice by changing its FC. But the principle you can see is that in the rituximab treated mice, what you see is about an 80% deletion within 16 hours of giving the antibody. So that's a nice system. It's quite a dynamic system, and it's rapid for assessing effector function. And one of the key findings that we made using this system was that if we repeat these experiments in gamma chain knockout mice, or if we repeat the experiment in mice that have been given clodronate liposomes, which are able to delete the macrophages and the phagocytes, we get no deletion whatsoever. And so to us, in this system with these antibodies, what it says is that the macrophages are the key effector cells in this model, and that they require the activatory Fc receptors. The flip side of that is the inhibitory receptor, and this is the seminal paper from Raphael Klein's in Nature Medicine at the turn of the century. And what it shows here is using two different antibodies, HER2 antibodies, that in a wild type Fc receptor 2 ++ mouse, that tumors aren't controlled as well as if you remove the inhibitory receptor shown at the bottom of the graph. So this fully supports the idea that macrophages basically mediate deletion through activatory Fc gamma receptors, and they're impaired by the presence of the inhibitory Fc receptor. So far, so good, and I think that kind of provides the background to how we understand kind of deleting antibodies. So the principle is then if we select the right isotype, our antibody should always work. And that clearly isn't the case when we consider what happens in the clinic. So this brings us to the question of why isn't that the case? And I think that's largely because pathology can change the rules. And explicitly in Southampton, we've studied the tumor microenvironment and how that can change the rules. And our real first inkling about how powerful that could be was demonstrated in the same model that I've shown you already, the adoptive transfer model. And the only difference to what I've shown you already is that we give a week before some BCL1 lymphoma cells. So relatively low number we give, and then we do exactly the same experiment. And what those experiments showed us that if we then still want to delete the human CD20 transgenic labeled B cells, that's much more difficult, and actually we don't get appreciable deletion at all using a very effective antibody that works perfectly well in the absence of tumor. We actually lose the ability to delete those CD20 transgenic B cells. So this isn't trying to delete the lymphoma, which might have other issues around it in terms of being good or bad at deletion. These are the perfectly normal human CD20 transgenic B cells. So this showed us how profound the tumor microenvironment could be, and that led us to start trying to understand what was happening. And to do that, we started to look at the tumor-associated macrophages that were present in these mice as the tumor was growing. So on day 7, 14, or 21, shown in the 3 different colors, what you see is a very profound impact on the Fc receptor expression profile of those macrophages that are associated with the tumor in the spleen. So Fc gamma receptor 1 and Fc gamma receptor 3 are going down. The inhibitory receptor, Fc receptor 2, is going up. And FCR4 is an interesting one that stays largely similar to how it does in the untreated mice. We've gone on and tried to understand the molecular basis of this, and we know that it's transcriptional. RNA-seq data shows that the profile that we see by flow cytometry, which is shown in the upper half of the panel, is mirrored exactly by the transcriptional profile. And we've been working hard to try and understand what are the regulators of those perturbations that happen on the tumor-associated macrophages. But the key observation really, I think, is twofold. One is that the increase in the inhibitory receptor and the decrease in 2 of the activating Fc receptors means that you have a very profound reduction in the activated to inhibitory ratio on these macrophages. So that's observation number 1, which would fit with the idea why they're no longer able to delete the target cells. But observation number 2 is that other cells that are present in exactly the same microenvironment, that's also coming from the spleen, monocytes and neutrophils, really aren't changed in terms of their Fc receptor expression profile, in terms of the dynamic behavior. So I think that's just fundamentally very interesting in how macrophages may well be more sensitive to these changes. And again, that's something that we're working hard to try and understand in the lab. Importantly, this isn't just a lymphoma-specific piece of biology. We've looked at a variety of different solid tumor models. Many of them I'm sure you recognize, those of you who work in tumor immunology. And in every case, what you see in the purple bar is the increase in the inhibitory receptor in the presence of the tumor compared to the level that you might see endogenously and certainly in the spleen. And again, what we see there is that there's an induction of the inhibitory receptor, which then suppresses the AI ratio shown in the bottom right in each of the 3 different tumor models that we've looked at. And Sergio Casada's group showed similar changes in Fc receptors in the presence of tumor in their immunity paper a few years back. So we think it's really important to study the Fc receptors in the context of trying to delete, and we've been doing that in Southampton in a kind of multi-pronged approach. We've been using structural biology, generating antibodies to the inhibitory receptor, trying to understand how it interacts with the receptor to antagonize or agonize the receptor, and that's something we've been interested in. When we can't get crystal structures, we're using molecular dynamic simulations. We've got a big investment in understanding the cellular regulation of those Fc receptors, and that's what I'm going to talk about next. And really trying to implement a whole suite of omics tools together to get a much more holistic understanding of how Fc receptors are regulated in health and disease. And often that means generating new mouse models to kind of test some of these hypotheses in vivo. So really what I've shown you is that the tumor has a very dynamic way of regulating the inhibitory receptor. And so the question came for the lab is what's regulating that inhibitory receptor? And our starting point for this was a paper we published a few years earlier, which was showing you that in the presence of high density, and high density here means 10 to the 7 per mil, as opposed to 10 to the 6 per mil, that monocytes in peripheral blood in PBMC cultures basically very highly upregulate the inhibitory receptor. You can see there in the red box, whereas the other Fc receptors might change a little bit, but nothing as profoundly as the inhibitory receptor. And this is an observation that Cam Hussain, one of our postdocs in the lab, made. And we were actually trying to replicate some experiments that Thomas Hunig had done, showing you that he was trying to find methods to predict what the Tigenero antibody, the anti-CD28, was doing. And we went back and employed some of his methods, and we showed that when you do that high density culture, what you do is you regulate the inhibitory receptor, which provides cross-linking and agonism for the anti-CD28 antibody. But as I mentioned, that's a story for another day. Again, in this PBMC culture, there are obviously lots of other immune cells present. But interestingly, much like we saw with the tumor microenvironment, we don't change the same receptor on a different cell type. So B cells aren't upregulating the inhibitory receptor any further, and NK cells, which are also present, aren't changing their Fc receptor in the presence of whatever's happening under high density culture. So we thought this was an interesting system to try and understand and dissect what's regulating the inhibitory receptor. And moreover, Cam did a nice experiment to try and understand really how profound those upregulations were. So we did the P-type numeration of how many receptors might be present on the cell surface. And what he showed is that when we measured a whole host of different molecules, that the inhibitory receptor actually became the most prevalent molecule at the cell surface, and obviously had a very profound impact on the AI ratio, as you can imagine. He went on to show that had a functional consequence, showing you that if you used red blood cells as targets for the monocytes and gave them anti-D antibodies, the low density culture, you still got permissive phagocytosis, whereas at high density that was reduced. And importantly, if you then added a blocking antibody to the inhibitory receptor, you could rescue at least a good proportion of that inhibition. So that got us to thinking about what's regulating 2B in these high density cultures. And we then obviously went to the literature and reviewed a whole host of studies that people have already published around different cytokines and different receptors that might be involved. And we collated that to a series of different cytokines that maybe we thought, or other immune modulators that might be important. And so our first port of call really was to look at those. And we did some microarray studies and looked at transcription to see whether over time in the high density culture, some of those previously reported cytokines might have been going up. And by and large, there wasn't a clear correlation with any of those receptors. We then said, okay, if we take recombinant molecules and put them in either at low density or high density, can we replicate what we are seeing in the high density cultures? So we just added soluble factors, recombinant molecules. So in the top graph, what you can see is taking low density cells and adding soluble factors. You can see that there are changes with things like IL-10 that you're upregulating the inhibitory receptor, but also some of the activating FC receptors. So nothing was really replicating what happens under high density, which is shown there in the column in yellow, which is very profound upregulation in the inhibitory receptor and not really for the others. We did the same experiment at high density and again showed the same thing, that we weren't able to replicate with the various cytokines what we were able to show with the high density. So none of those soluble factors, known cytokines, seem to replicate what we do and observe at high density. We then did a pretty exhaustive R&D experiment, which is an uncategorized, a very clearly categorized failure, again, using recombinant molecules from a whole host of studies that had reported potential reasons for modulation, and none of them, again, modulated what happened with high density. We then started to question whether or not this was transcriptional and showed it very nicely was, using actinomycin D to show that we don't get this upregulation of the inhibitory receptor when we block transcription, and we used various other epigenetic modulators like azacitidine and DZNEP, and showed that that wasn't sufficient. So reversing those methylations was not a way that we could upregulate the inhibitory receptor. So it's transcriptional, not due to methylation. And so at this point, really, it was still a very much an open story as to what might be regulating the inhibitory receptor. So we went to the omics and considered the transcriptional changes that we see after high density and get the usual story, like thousands of genes going up, thousands of genes going down. And then we started to look at them a bit more closely to try and understand what's important in the regulation of FC receptors in high density monocytes. And that study was really useful because what it showed us was that the high density culture over 2 to 10 hours was clearly able to upregulate the transcription of the inhibitory receptor. And it mimicked a data set that we'd come across, which was a hypoxia data set by Bosco et al., published a couple of years before. But when we started to look at our top 50 regulators, it was also abundantly clear that many of them were actually modulators of hypoxia. So HIF1A, the top one there, ARNT, VEGF, a whole load of molecules that you can see part of the HIF-mediated hypoxia signaling pathway were all clearly involved. So the question then was, are we indeed seeing hypoxia? So we started to measure upregulation and stabilization of HIF. We were able to show that that was happening. CA9 and GLUT1, two of the downstream reporters of HIF and hypoxia, again, were regulated on the monocytes in these high density cultures. This then brought us to the question is, is hypoxia actually being explicitly observed in these cultures? And to do that, we used this very nice OXO dish, which enables you to measure oxygen levels in your cultures. And when we did that, it was very clear that culturing cells either at low density or high density, very rapidly, within half an hour, you are making the cells clearly hypoxic. They're dropping below 2%, 1% oxygen, and that persists for the rest of the culture. So that gave us a very clear impression that hypoxia was indeed being induced, and it was being induced very rapidly. So the next question was whether we could use hypoxic stimulation to upregulate the expression of 2B outside of the high density culture. So we did this in two ways. We used a hypoxia chamber, physiologically limiting oxygen below 1%. And we also used various prolyl hydroxylase inhibitors such as DMOG or roxadustat, as you'll see later, which basically inhibit those enzymes, which are then able to mimic physiological hypoxia. And we saw what we'd hoped, which is, as you can see in the flow cytometry plots at the top, High density cells under without any change in oxygen levels, you see that very profound upregulation of the inhibitory receptor. You do low density cells in a hypoxic chamber, and you get very high levels of upregulation. And the same is seen with the DMOG. And that's just shown there for multiple donors. It also, we were able to use siRNA to knock down HIF in monocytes very nicely, and we were able to show that that completely blunted the upregulation of the inhibitory receptor, either using high density or DMOG cultures. And again, multiple donors shown to demonstrate that. But it didn't tell us how it was changing 2B, and so to do that, we started to think about how HIF might be recognizing 2B, and we looked in the promoter of 2B, and there's no hypoxia response element for HIF to bind directly. So we considered then indirect methods, and certainly there is an AP1 binding sequence within Fc receptor 2B, which can be made up of JUN and FOS. We went back to our potential regulators that we'd seen in the transcriptional data, and you can see here that AP1 and JUN were indeed in our top 50 regulators. So again, we went down the siRNA approach. We knocked down JUN and showed that we could no longer then get upregulation of 2B, either under high density or under DMOG stimulation. So supporting the role for JUN, again, multiple donors. So the summary here is that we think for high density monocytes, they do indeed become hypoxic. That stabilizes HIF, and you get the upregulation of the downstream molecules. And that we get upregulation and activation of AP1, which can then drive the upregulation of the inhibitory receptor on the monocytes. So monocytes obviously are the precursors to macrophages that can infiltrate to infiltrate and then differentiate. And so the question was, does the same thing happen for macrophages? And in essence, I wouldn't be talking about it unless they did. And what you can see here, I think very nicely, again, under normoxia and under DMOG stimulation, we get this nice upregulation of the inhibitory receptor. Again, for multiple donors, we see the same thing under physiological hypoxia in a hypoxia chamber. We can do the same thing under differentially stimulated macrophages, so using LPS interferon gamma or IL-4, IL-13, and using roxadustat, just another molecule very similar to DMOG, and again show that we get upregulation of the inhibitory receptor. Interestingly, whether hypoxia affects 2B in the same way in macrophages is something that we're studying quite intensively at the moment. It looks like there are differences in the molecular basis for how 2B is upregulated in macrophages, and that's a work in progress at the moment. But the key question is whether this is observed in vivo and its functional relevance. And so to address that, we made use of roxadustat, which is another prolyl hydroxylase inhibitor, and we use that because it's more stable and potent in vivo. We gave it IP, and then we looked at the macrophages in those, either in the lavage in the IP cavity or in the spleen. And what you can see in the red indicated areas is that under roxadustat treatment, what you see is an upregulation of the inhibitory receptor on the macrophages in both of those compartments. So we're able to elicit a degree of hypoxia in that particular niche, which then we could explore whether that had a functional impact. We were also able to see whether this happens physiologically in tumors. So some of these tumors you've already seen, mentioned of in terms of enumeration of upregulation of 2B in the presence of the tumor on the tumor-associated macrophages. This was to see whether the increase in 2B was related to areas of hypoxia, so microenvironments within the tumor itself. So we used hydroxy probe to assess that, and what you can see is a nice coincidence, hopefully, between the green and the red, and in essence showing you that the very high level of regulation of 2B entirely coincides with hydroxy probe staining. And we did that in 3 different tumor microenvironments. But the real proof in the pudding is whether or not this has functional consequence. And to address this, what we did is we used the same drug, roxadustat. We gave that into the peritoneal cavity. We then gave some EL4 CD20 positive tumor cells. Then we gave rituximab to try and delete them. And essentially, we were assessing whether the presence or absence of hypoxia that was induced by roxadustat was able to prevent the deletion of the tumor cells. And that's exactly what we saw. So some raw flow data on the left showing you where the tumor cells are in the red box. With Ritux, we're able to delete them. When roxadustat is there, we are no longer able to delete them. And that's just enumerated in the graph to the right. So the first half of the talk, hopefully I've been able to convince you that direct targeting antibodies deplete through activating Fc gamma receptors on macrophages. Inhibitory Fc receptor 2b mediates resistance. Tumors can upregulate 2b, and that can be through cytokines and or hypoxia, and there are other mechanisms too. And that's driven by HIFs and AP1 in monocytes, and as I mentioned, it may be more complicated in macrophages, but that all elicits a lower AI ratio, which reduces antibody efficacy. But really, how does 2b inhibit that efficacy, and what's the best way to block it? So this is the second part of the talk, and this is really when we start to consider the integration of ITAM and ITIM signaling to modulate those antibody effector functions. So on the top is the macrophage. You can see engaging with a target cell, and it basically interacts through the various Fc receptors. If there's no inhibitory receptor, then we stimulate the ITAMs, we get downstream signaling, we get ADCP, that causes target cell deletion. However, if the inhibitory receptor is there at the same time, what we then we get is activation of the ITIM, phosphorylation, and we get SHIP and SHP1 that can shut down that signaling, reduce ADCP, and promote effector cell inhibition, or at least that's the paradigm that kind of exists. And we've been working on blocking the inhibitory receptor for a little while, and we were interested in this concept. And it was clear to us from doing this work that blocking the inhibitory receptor with antibodies is very different than having a genetic knockout, as had been done in the classical Raphael Klein studies and in others. And this came from some work that we published alongside BioInvent International, where we generated some fully human antibodies and shown that blocking the inhibitory receptor was able to potentiate rituximab-mediated deletion of B-cell targets. And we proposed a variety of different mechanisms. One thing that happens with rituximab is that it can internalize from the cell surface, and that process is actually driven in part by interaction of the Fc of rituximab with the inhibitory Fc receptor on the target B-cell surface. And the antibodies, the 2b blocking antibodies, are able to stop that process. The 2b antibody can also bind as a target deleting antibody itself to the 2b positive B cell. And also there's the potential for the 2b to block inhibitory signals coming into the macrophage. And we were just interested about which of those mechanisms might be more important, and whether we could engage or ameliorate those with different Fc backbones of the antibodies. And so in essence, we developed two different formats. One was a wild type format, which can bind 2b through its Fab regions, and then all the other Fc receptors through its Fc, or an NQ version, so a deglycosylated version, which can't interact or has very little interaction with Fc receptors through its Fc, and really is only able to block the inhibitory receptor itself. We've also got antibodies that bind to the mouse inhibitory receptor in exactly the same way, so wild type or NA versions of that molecule. And so this is work that Ali Reganian in the lab did along with a Ph.D. student, Alex Simpson. It was published last year in Cell Reports. And so what we wanted to do is really separate out the importance of blocking the inhibitory receptor on the target or on the effector cell. So setting up various cell systems where we were able to do that using a human Fc receptor 2b transgenic mouse we developed a few years earlier, crossing that onto the human CD20 transgenic, and then modulating whether or not we have the inhibitory receptor there on the target or the effector cell. And then using the relevant blocking antibodies either as a wild type or as an NQ. And when you do those experiments where you only have the possibility of blocking the inhibitory receptor on the target, what you see is rituximab, as we go from left to right, rituximab gives you a certain level of deletion in our same target to non-target deletion assay of normal human CD20 transgenic B cells. You get a certain proportion of deletion with rituximab. Using a wild type blocking antibody, again, gives you quite nice deletion. There's reasonably high levels of the inhibitory receptor on these B cells. An NQ antibody on its own doesn't do anything. It's got no effector function, but the combination of the two wild type antibodies gets you even better deletions, additive deletion, but that isn't shown to be the case when you use an NQ version of the inhibitory blocking antibody. So that tells us that a wild type Fc blocking antibody that interacts with all the Fc receptors works best in the context of just blocking the target. If we then just block the effector, what you see again is a proportion of deletion with the rituximab. Neither the wild type or the NQ blocking antibody do anything, because in this context, obviously, there's nothing on the target. If you then do rituximab plus a wild type antibody, it actually gets worse than Ritux on its own. But if you add an NQ blocking antibody in this context, you get better deletion again. So clearly here then the NQ antibody that interacts only with the inhibitory receptor works best. And we started to think about why that's the case. And the obvious place to look at was because the inhibitory signaling. So there's an ITIM and you get an inhibitory signaling into the macrophage, and that's why you shut down phagocytosis. And certainly we were able to demonstrate there was indeed phosphorylation of the ITIM when you use a wild type but not an NQ antibody. But was this actually all that was happening? We thought about, okay, well, let's go back to basics and consider the possibilities. And so what we did is we looked at the possibility that there was differential ITIM signaling, as I just suggested. Possibly there's an altered competition for Fc gamma receptors. And the question then came in therapeutically, do we need to target the ITIM signaling or just blocking the inhibitory receptor interaction, or both? And to do that, we need an in vivo model. And so this is the model that we demonstrated. So I'm not going to go into this in any detail, just essentially to show, to tell you that we knocked out 2 tyrosines in the ITIM in this mouse. We demonstrated it could no longer, the ITIM couldn't be phosphorylated. We showed that the ITIM had reduced functionality in terms of internalizing immune complexes, and also that the ITIM lacking mouse, which we call the no-TIM, basically was not able to suppress signaling through the inhibitory receptor, which is shown in some calcium flux assays there in the bottom. So just in the interest of time, I'm not going to go through this in any great detail. It's all published, it's all detailed in the paper. But the key take-home experiment was essentially this one, which was taking 4 different mouse models. So a wild-type mouse expressing only mouse Fc receptors, a mouse lacking the inhibitory Fc receptor, the endogenous mouse receptor, the same mouse then with the human Fc receptor as a wild-type molecule being put back in the context. So the only inhibitory receptor in that mouse was the human one, not the mouse. And then the same background again, but this time doing it with a no-TIM signaling context. So no signaling through the ITIM in this particular mouse. So the real key question and the main conclusion was, is there improved in vivo target cell deletion in the 2B signaling mutant? And so the experiment was very simple. We used the CD20 deletion model again, but we did a sequential increase in the amount of antibody we gave. So 2, 10, or 50 micrograms, then we bled for B cell deletion, and then we were able to see whether or not the different contexts of those different mice, the 4 different mice strains I mentioned, were able to impact what was going on. And the preliminary experiments essentially went according to plan. So a wild-type mouse, as you give increasing amounts of the antibody, you got more deletion. If you knock out the inhibitory receptor in the mouse, you got better deletion, which is the purple line. If you then put in a 2B transgenic from the human, you then get worse deletion. So all of this was as expected. And then the question was, what happens if you put a 2B transgenic with the no-TIM, so no ITIM signaling? And it went completely different to what we anticipated. So if the ITIM was important for suppression, it would have gone down to where the blue line was. But actually what we saw was that this mouse was actually even more suppressed in terms of its deletion rather than less. So this pretty much said that the inhibitory signaling through the no-TIM for the ITIM, is not important for resistance to the CD20 deletion. We considered some potential reasons for this. One possibility is that the no-TIM receptor might be driving rituximab internalization differently, and we didn't see that that was the case. Another possibility is that there may be different half-lives of the antibody in vivo in the different mice, and again, that was shown not to be the case. And then the next question was whether this was due to an impact on the target cells or the effectors. So we may have been changing impacts on either of those populations. And to address that, we essentially did the same trick as before. We took systems basically where we're only considering the inhibitory receptor either on the targets or on the effectors. And so in the first case, when the 2B is only on the target cells, we were able to show that we got nice deletion. So having a no TIM on just the target cells didn't impact the ability for those B cells to be deleted. If we then do the reverse with the 2B only on the effector cells, what we were able to show is in a system where it was in a knockout, we got a nice level of deletion. If we put the no TIM onto the effectors, then we got a suppressed level of deletion. And that was very clear. So this was the suppression that we were seeing. So basically having the no TIM 2B on the macrophage was the important part. And moreover, the question then was whether that is simply because it's interacting with and competing with the activating FC receptors for the FC of that deleting antibody. So to consider whether that was the case, we again came back in with a blocking antibody to the 2B, which remember is only on the effector cells. And what we were able to show, I think very nicely, was that if you then block the 2B, you go down to the level that would be seen on a knockout. So you completely reverse the suppressive phenotype. Excuse me. And so what do we think is happening? We think essentially what we're talking about here is not ITIM signaling being the suppressive mechanism, but essentially just competition for FC. That in the context of having high levels of 2B, whether that be a no-TIM or a wild-type molecule, it's able to bind the FC, which stops the FC binding to the activating FC receptor. If we then block the inhibitory receptor with an antibody, it then frees up the FC to bind to the activating FC gamma receptor to trigger signaling and then deletion. And we think this is essentially just recapitulating the scorpion effect, which had been reviewed back in 2012 by Mark Hogarth and other authors, which was described as, say, the scorpion effect or the Kurlander effect, where the FC, the Fab's binding at the cell surface in exactly the same way you anticipate, but the FC is able to then interact with other FC gamma receptors to cause their blockade and occupy them. So we think that's one of the reasons why these different antibodies are able to delete well or not so effectively. So just to summarize and conclude what I've hopefully told you about this evening is that the direct targeting antibodies delete through the activating FC receptors on macrophages. Inhibitory 2B can mediate resistance. Tumors can upregulate that 2B, which elicits a lower AI ratio, but that inhibition does not seem to be independent on ITIM signaling. I haven't shown you any tumor data, but there's tumor data in the paper. There's also Treg data in terms of deletion. So this isn't just a CD20 B cell phenomena. This seems to be across the systems that we've tried it on. But importantly, effector function can be enhanced in vivo by blocking antibodies. And I've shown you the context of those blocking antibodies, whether they're themselves FC functional or FC blocked, is important depending on what the mechanism you're trying to engage. And when targeting the myeloid effectors with antibodies, we've got to consider the potential of that FC gamma receptor blockade and competition, because obviously cells like macrophages have multiple FC receptors, and we need to think about how that might impact our different biology. But for 2B targets, we think a wild type antibody, which can augment target cell deletion, might be most effective. And for 2B negative targets, that an FC null antibody is probably optimal to block effectors. So I'll just end with the acknowledgements. So all of this study was done in close collaboration with Stephen Beers, Ali Reganian, and Bjorn Frendeus. Cam and Alex were the two kind of lead people on the ground doing a lot of the work. We have lots of clinical collaborators that have helped with this. This is a collaboration also with GSK. And hopefully I've acknowledged all the funding that was used for these studies, and just to acknowledge the fantastic collaboration we have with BioInvent on the 2B antibodies. So I'll stop there and take any questions.
Thanks, Mark. That was awesome. Maybe I'll get started. I don't see any coming through on the chat, but maybe I'll get started on some questions. You showed a slide. It was the AI ratio over time on the macrophages. And I noticed for 2B and a couple of the other that there were groups of outliers of mice What do you think that is? Do you think that the tumors are different? Do you think the MACs are different? What do you think it is?
So I think some of it comes down to tumor size and how quickly. So really early on, you see actually an inflammatory response because you're doing an introduction of the tumor. So you quite often see a blip early on, and then you start to get this progressive tumor growth. But certainly within individual mice, the tumor size, the splenic mass can be quite different. And when you normalize it for splenic mass, then things tighten up quite a lot. So yeah, it's interesting. We've done a few other lymphoma models, and the BCL1 seems to have the most profound effect. A20 and some of the other models show similar effects in terms of it tends to be 1 and 3 go together, 2B goes up, and FCR4 is really interesting because it doesn't seem to be impacted in the same way as the others are.
Interesting. And your antibodies that you had, I mean, it was great to have the different mice, so you didn't need this, but do you have 2B specific antibodies?
Yeah, so those are 2B specific.
Those are 2B specific, because only like 2 amino acid differences or something like that between
it's
right?
90, I should know this, 95% identical in the extracellular domain between 32A and B. So I was always skeptical that BioInvent would be able to make them, but they indeed did. So we spent a lot of time characterizing and testing them, and so did they. So that all of that characterization is in the Cancer Cell paper. And yeah, we've actually solved the structure for some of those antibodies. So we know where they bind, and we know why they're specific. And it is an amazing feat of, specificity that the 1 or 2 amino acids can determine, AB selectivity, not just selectivity. They either bind or they don't bind. Whereas some of the earlier antibodies that were reported to be 2A selective, as IgGs, not in our hands anyway.
Yeah, I agree.
Yeah, good.
It's really interesting. I wonder what would happen if you included the 2B. So because we're specifically interested in SARS-CoV-2, I wonder what would happen if we included that with some of the other antibodies that have effector function monoclonal, not as bispecs, but as combos.
Yeah, no, I think it's really interesting. I mean, there's a whole kind of body of literature coming out now, which is, we always thought that we understood deletion at least. And it turns out that maybe we don't understand deletion quite as much as we thought. And actually there is probably signaling back into the myeloid cells that's actually changing their transcriptome and kind of behavior. There's been a couple of recent papers that support that. There was some nice paper by Jeremy Waite a few years ago, looking at this suggestion that actually things like CTLA4, which can delete Tregs, looking at some other things that might delete Tregs, then looking at kind of immune stimulation. It's not just deletion that is doing that. It's some reciprocal signaling in some of those myeloid cells and reprogramming and rewiring. And I think in COVID, there's some amazingly complex biology going on, right, in terms of inflammation and what's really going on there. And the suppression of Fc receptor signaling in different contexts, I think, that would be interesting. It's just really difficult to model, the complexity of that, to say confidently, yes, we should block the inhibitory receptor, and if we should block it, how should we block it without exactly. And is it too late by the time that you see a patient that's already quite sick?
So it looks like one question came through. Can you overcome the FCR competition by giving more antibody?
That's a really good question. Someone asked me that the other day, and we ended up in quite a philosophical discussion. It was around dosing in general, about therapeutic antibodies. And my feeling is no. But then I'm kind of always aware of these to me, quite strange things that people do in radio immunotherapy, where they give a cold shot to block what they call non-specific sites, and then they go in with the kind of radio-labeled antibody. And I've never understood how that works, why that works. So again, in this kind of thing, could you overdose? Would you? I don't know. Surely a mathematician should be able to tell us these things, right? In terms of modeling the receptors. And
I don't know. That might be a little difficult. And I also wonder how age affects all of this as well, and how you might have to to do things that are differently in the elderly versus not?
Yeah, I think there's been a lot of antibody kind of combination therapeutics. So I'm thinking of the PI3 kinase delta combinations with antibodies that in second or third line, they were fine and they were really looking quite good. And then they tried to drop them down into healthier people. And then they started to see toxicities they hadn't seen. So I completely agree. It depends where you are on that threshold of where your immune system is when you start poking it around.
Two other questions. One from James, I'm not sure which James. When designing effector function assays, specifically ADCP assays, given this data, should we be giving more consideration to effector target ratio to avoid hypoxia or even induce it for a full overview?
Yeah, no, that's a really good question. You know, when we did some of our studies, we were thinking, you know, should we actually perform these experiments under hypoxia? And, you know, technically it's difficult to do some of those things. Target, I think in those kind of assays, and I don't know, it depends how long your phagocytosis assays are. Ours aren't run long enough for them to be a consideration for hypoxia. The number of cells, we certainly wouldn't see an issue with that. Not to the impact that by the time you've done the phagocytosis assay, you might be starting to read out hypoxia, but you've already done your phagocytosis by then. But if you're doing certainly long-term cultures and measuring phagocytosis, then certainly it would be a consideration. Yeah.
And then Tao asked about FC gamma R2B1 and R2B2.
Yeah.
Do you think both subtypes do not purely rely on ITIM, as you only showed 2B by macrophages? How about 2B on B cells?
No, really good question. So the transgenic that we have is 2B2, but it still has its inhibitory function on B cells. So even though it's the wrong one in terms of it doesn't have the additional strength, it still does inhibition. So it's ITIM functions, and then we use exactly the same construct and just knock out the tyrosines in there, and then it doesn't block calcium flux. So the calcium flux experiment I showed and I didn't explain very well is a B cell assay. So that's B cell receptor stimulation, calcium flux being inhibited by the ITIM in the transgene that we put there. But it's a really good question. I think the 2B1, 2B2 is still very, very interesting in terms of the differential biology. And I think the ITIM may well be much more important for B cells than it is for macrophages.
Interesting. Yeah, sure. There's a question. I'm sitting in a room with 10 people, Mark.
Okay.
So I'm not sure if you'll be able to hear her, but here's a question. Here she comes.
And did you try different level of hypoxia in different model? For example, like 2 solid tumor model by MC38 or B16, or these are 2 different solid topical solid model, or like B16 can be used to set up a topical solid tumor and also metastasis model. When you compare the 2 different models, did you see a different level of hypoxia, which in turn caused a different reduction of 2B or the percentage of 2B positive cells?
Yeah, no, really good question. So we haven't done those experiments. In vitro, we've looked at varying the amount of hypoxia. So 0.11, 2% oxygen. As soon as you drop below kind of 2%, you start to see the impacts. And there's not like an increase past that. It's a kind of response once you're into hypoxia that you see anything under kind of 1%. So you don't see an increase of 0.1%, for instance. In terms of the different tumors, definitely different tumors, individual tumors certainly, but different types of tumors certainly will see more or less hypoxia. But we've never done that. That's a nice question, actually. Yeah, in different sites, are there different levels of hypoxia, and does that correlate? We haven't done that. I mean, Folk's talking to you in a couple of weeks, right? So he's done much more in different environments looking at macrophages. I don't think he's explicitly looked at hypoxia, but yeah, it's an interesting question.
I think this will be also very interesting in respiratory infectious disease.
Yeah, sure. Absolutely. I mean, we know in the bone marrow where that's more of a hypoxic environment, pleural cavity, things like that, we see a higher level of FC receptor 2 in the mouse models and in clinical samples.
Thank you.
Yeah, awesome. Thank you.
Thank you so much, Mark. That was really interesting and a lot of work with all those different transgenic mice and knockout mice.
Yeah, Alex did a good job.
Yeah, it was fantastic. And a really good overview of definitely 2B, which we don't talk about enough, by the way.
Yeah.
For sure. All right. Thank you so much, everyone. We're at time, but looking forward to seeing everyone again in a couple of weeks when Folk gives a seminar on the 23rd.
Cool.
Thank you.
Thanks a lot.
Bye-bye.